Hello, students. Welcome to my second screencast here for your biochemistry test coming up. This is Mr. Gibney. We're going to go over macromolecules, so bigger molecules, right? They're still small. They fit within a cell or they help to make up cells, but they're bigger than individual atoms, and they have emergent properties. They have functions that promote life. So carbohydrates providing energy, lipids helping to store energy and provide cell membranes. Proteins do everything, right? They serve as enzymes, structural supports, etc. And nucleic acids serving as your hereditary information, which you can pass on. Just like all my videos, this is not for profit. This is labeled as Creative Commons, so it can be shared and used and hopefully help students. That's what it's made for. I'm going to use some of my own images and some images from Khan Academy and OpenStax, and those are available for free on khanacademy.org or OpenStax. And if any images are copyrighted, just let me know and I'll take them down. All right, here's my boy during our break that we just had in Kaloof, and he's happy because he just got to eat some carbohydrates. He was having a blast up there in the sand dunes. And then we finished it off with some lipids, having some fun at the Chetty. Okay, this unit, or this part of the test, tends to stress students out the most. And if you're watching this video early, don't worry. We're going to do a nice graphic organizer on a poster paper leading to the structure and function of molecules. Also, you should review the POGL that we did in class on biological molecules, and hopefully this video will help you. Also, remember we used a lot of the modeling kits in class as well. So I'm not going to ask you to remember complex chemistry when regarding the structure of certain molecules. Right? This is an intro ninth grade biology class. So I just want you to know about it in general and then the functions of each macromolecule group. And I hope you can kind of pierce it together and kind of see some overarching connections with ecology and evolution, right? If a protein changes, maybe it has a different function. Maybe that function could be something that's selected for, etc. And it could be something that is that you can see an interdependence among organisms. All right, let's bring it back into what's in this test with biochemistry. So you should pause the video and see if you can answer these questions and model these polymers and describe the functions and structure of each. If you can, then you can skip the rest. There's no need. If you can't, then let's learn it together and let's go through it. And I bet you know more than you think you do. All right, so here is a chart that goes over what each macromolecule is, some examples, definition, what they do, where you find them, etc. So we did a fun lab where you got to test for proteins and starches or carbohydrates. And you figured out that iodine changes colors in the presence of starch. And Barrett's reagent changes colors from blue to a beautiful purple in the presence of proteins. And we tried to reinforce how we conduct science experiments with that. So I hope you enjoyed that lab. And then if you're watching this early, we're going to do a lab with enzymes. If you've already done the lab, then hopefully you've seen we did a lot of work on how what environmental factors affect enzymes, right? And whether or not enzymes work. And we did that with catalase, with hydrogen peroxide. Okay, let's dive in. And I'll get a lot more into enzymes with the next video. So can you remember the function of a carbohydrate? Can you tell me how the structure of that carbohydrate contributes to its function? So you probably remember the first one. The second one is hard. All right, I don't want you to be off put by it. So let me give you an example to kind of show you how to break it down, right? First of all, the function is quick energy, right? This provides energy for our body. We can break it down and turn that energy or use that energy to make ATP, which is energy for our body. So let's think about the structure of it now. We know the function, quick energy. The structure, well, it's like this ring-shaped molecule. It had carbon, hydrogen, and oxygen. It had them in a certain ratio, which was way different than lipids or fats, right? It had carbon, hydrogen, oxygen in a 1 to 2 to 1 ratio, slightly different in the polymer state, but it had a lot more oxygens than a fat. And so now let's see a more vivid example, right? There can be different types of carbohydrates. You could have starch, like a carbohydrate in a potato or pasta, which you probably think of as carbohydrates. And then you could also have cellulose or tree bark, which is made up of the same thing, the same glucose molecules, but yet it has such a different function. It's providing structure and support. It's not something that you could eat, although some of you pointed out in class that you could eat tree bark to survive in the wilderness. Just say in general, you can't eat cellulose, right? But starch you could. So let's see the difference between them. Well, here is starch, and these are between glucose molecules, and you can notice, wow, they're all facing the same direction. Whereas in cellulose, you got up, down, and this is how the different structure of the molecule, all up, down, the different structures lead to different functions. Because these are all stored in the same direction, they can be broken apart and used for energy. So this is starch. Whereas cellulose, this is a structural support. All right, so hopefully that helps key you in on how the structure of a molecule can contribute to its function. Remember, I'm not going to be really hard on that in this test. Just want you to appreciate in general. So some quick information about carbohydrates here is glucose. You should probably use that in a promposal. Isn't it sweet? C6H12O6. Well, if you link a lot of them together, then you would get a formula that looks like this, C6H10O5, and this is due to dehydration synthesis. All right. They provide quick energy. Plants make them through the process called photosynthesis, which we'll learn more about. They're fruits, vegetables, and grains. A tip for you, if you ever see a word that ends in OSE, it's usually a carbohydrate. Lactose, fructose, sucrose, maltose, etc. Okay, so a little more in-depth notes. Mono means one, saccharide means sugar, so one sugar. These would be simple sugars or the building blocks. Whatever is a monomer is the building block. Poly means many, so this is the when a bunch of them are joined together. So a disaccharide means two sugars or two monosaccharides linked together by a process called dehydration synthesis. Just like it sounds like dehydration means to take out of water and synthesis means to put together. We have a fancy name for that called glycosidic linkages. All right, and so an example of this would be maltose and sucrose. Polysaccharides means many sugars, many of them linked together. It can be hundreds to thousands. So you got your storage ones like starch, and we would use glycogen in our liver to store glucose. And energy can be released by hydrolysis. This means hydro means with water, and lysis means to break. Structural polysaccharides like the cell walls and bark, cellulose, chitin, the exoskeleton of insects, spiders, and lobsters, etc. All right, and so let's move on. Here's just an example of monomers and polymers. So monomers would be the individual building blocks or the individual Legos, and the polymers when they're all put together. And if you arrange the monomers in different ways, you get different polymers, whether it's a bridge or the tower or whatever that my kids love to make with the Legos. It's the same Legos. They're just arranging them in different structures, which lead to different functions. All right, so hopefully that helped you with structure and function. Remember I told you carbs are the trickiest. So once we get through these, the rest should be a little easier. So here's an example of dehydration synthesis. All right, we're going to have to use energy to put these together. We're going to have to use an enzyme. Here we're going to take off this H from this OH. We're going to take off this OH here. And then that's going to become a water molecule. And we're going to bond on this O, and that's called an oxygen bridge. It's a glycosidic linkage. So here's the resulting molecule, right? And so this would be an example of how the starch is made, right? They're all facing in the same direction. So large molecules, we could break it apart by inserting the water and using an enzyme. That would be hydrolysis. All right, carbohydrates, did you get the function? Quick energy. You get the structure. So they're a 1 to 2 to 1 ratio of carbon, hydrogen, and oxygen. A little different for the polymer, right? It's can be used for structural purposes as well. So what's a big difference from lipids that we're going to encounter later on is they have a lot more oxygen. Okay, so we started with carbohydrates, which is tricky, but now we can tie it all together with lipids. So what are they? What are the functions of the following lipids? Saturated and unsaturated fats, phospholipids, steroids, and hormones. And what structures lead to the functions listed above? Can you do that? If so, then you can fast forward through the lipid section. If not, let's relearn it together. All right, lipids are fats, waxes, steroids, and phospholipids. They're items that are non-polar or do not dissolve in water. I showed you that at TED Ed where the oils couldn't join the square dance. They just can't get in there. It's not energetically favorable. So they help to insulate us. They help to keep us warm. Not something we need too much of here in Omaha, but maybe on a desert trek when it gets cold at night. They do provide energy, right? We can break down fats for energy. And they also help to serve as cell membranes that are really important to us. Whoa, check this out, right? They're also made of carbon, hydrogen, and oxygen, the exact same elements as carbohydrates. They're just in a different shape, which is a different structure, which leads to a different function. All right, and they store more energy than carbohydrates. Okay, so a major theme is structure leads to function, and this is just talking about that. The arrangement or structure of the carbon, hydrogens, and oxygens and carbs make it easier to break up, while fats, they form long chains which store more energy, and they don't have as many oxygens, so they're going to need to oxidize more to get down to CO2 and water. Okay, I'm going to take a quick break, and then we'll come back for... Okay, thank you. So lipids, here, let's go over some fats, right? And these are called triglycerides. You've probably heard of that before. It's a glycerol molecule that then has these long fatty acid chains, right? And these chains have all these carbons and hydrogens, and they store a lot of energy. If it has a double bond between two carbons, then it's going to have a kink. And those double bonded ones are unsaturated fats, and they'll be liquidy like olive oil. And saturated fats have no double bonds, and for example, that would be butter. So some people argue about which fats are better to eat. In my opinion, it's just we probably eat more fats than we need. So that would be something to check on there. All right, the actual linking together of the fatty acid chain and the glycerol molecule is once again a dehydration synthesis reaction. You're going to kick off this H and this HO here, and that's called an ester linkage. Here's unsaturated, saturated. Trans fats have hydrogens added. So let's see an animated GIF. Let's see it happen. So here's the fatty acid, and here's the glycerol. Here it's going to use an enzyme to kick off that water molecule, and now it's going to combine here on that oxygen, and that's called an ester bond. All right. Here we can see it breaking it. So let's say that you're running, you're doing a lot of exercise, and you're going to burn off some fats. This is what's happening. You're breaking off that chain, and then you're going to oxidize or break down this whole fatty acid chain. So when you lose weight, you actually lose it as carbon dioxide that you breathe out and water that you sweat out and pee out. Okay, those are fats. Now let's talk about cell membranes. So they have the fatty acid tails. They have the glycerol molecule. But look, they have something different. And that different item is called a phosphate. And that different item is negatively charged. So now we see the different structure, students, leading to the different function. And what is their function? Is they're going to serve as cell membranes, right? And so they'll naturally form this, which is pretty cool. All the parts with the negative phosphate will face outwards towards the water. That's called hydrophilic or water-loving. And then all the parts that fear the water, which are these nonpolar tails, face the inside. This is energetically favorable, so they naturally form. And so we have cell membranes. And membranes are a big thing. They lead to emergent properties, right? You can have what are called membrane-bound organelles, like the mitochondria, the nucleus, lysosomes, etc. And they can all do different stuff because they can have different pHs on the inside. They can have different proteins. They can have different functions. So membranes are a really big deal in biology. All right. These we didn't really get into. Okay, so steroids, things like estrogen, testosterone, cholesterol, etc. These types of hormones are classified as lipids. They don't look like these. They have these ring shapes, these four fused rings. So why are they lipids? They're lipids because they're nonpolar and they can cross easily into cells, and then they can make things happen. This is pretty cool, though. You'll see that they have a similar structural backbone and in different little functional groups. And those different little functional groups make them have different purposes. All right, so that was lipids. Okay. So you can hear them yelling downstairs, and I have limited time to make this, so we're going to plug on. Hopefully they're not being too loud. We'll go through proteins. All right, we were at the car wash. She was waiting for her egg, which would be protein from the coffee shop, right? You tested those egg whites with Barrett's reagent, and they turned purple. So can you do these? What are the functions of proteins? What's the structure of a protein? How does the structure lead to its function? If you can, then skip this section. If you can't, then let's relearn it together, and you're going to get it. All right, proteins are a bunch of amino acids put together. That's really important. The amino acids are the monomer. The polymer is the whole protein. They do everything. When you're looking at me, you're seeing proteins. They're doing enzyme reactions in your body. They're building muscles and bones. They help to fight infections. All those coughs that I heard as we were coming back from this break, and myself included, right? I have proteins or antibodies fighting it. All right, they're made of carbon, hydrogen, and oxygen, the same elements inside of lipids and carbohydrates, plus one more, nitrogen. Now, some proteins can also have sulfur in them, and some of them can also have selenium, which are due to different R groups. Okay, let's go over the structure of an amino acid. So here's the amino group, and here's the carboxyl group. If you know anything about pH, this can give off this H plus proton. So therefore, this is the acidic part. This can accept one. So this is the basic part. So an amino acid, right? So here we go. The amino acids are all the same backbones. They have different side chains. So a good example of that would be that you all have to wear the TASM uniform. So you all have this, but yet each of you are different because you customize yourself. You have different haircuts. You wear different shoes, different stuff. Those would be your side chains. This would be the backbone, would be what you're supposed to wear to school, the TASM polo and regular pants, and then the side chain would be the differences. What are some other examples of that? Thank you. Sorry, I had a cough. All right, so some other examples would be playing cards, right? They all have the same backbone, right? They all have the same plastic, etc. Oh, I got to get the plug. Okay, and so sorry about that. And to come back, so for example, maybe cars, right? They all have the same under chassis system with similar engine structure, but then their outsides look different. That would be the side chain. So the primary structure is the order of these amino acids. And we'll get into that when we do our DNA to protein unit. We'll get into how does DNA code for that. So here's an example of an amino acid. Here's some examples of different R groups. So lysine, glutamine, aspartate. Maybe these sound familiar, right? You get them from the foods you eat. You can also make some of them. That's why it's important to eat more than just pizza. Look at this. They're formed the same way. This is through dehydration synthesis. They take off the OH from this. This is called a carboxyl. And they take off the H here from the amino, and that forms water. And now you have your peptide bond or your polypeptide chain, many bonds together, right? So now the amino acids are linked together. Remember, the R groups are going to be different. And those R groups are going to have different structures, which will lead to different functions. So here's an example of a GIF of that happening. So that is our peptide bond. Here is hydrolysis breaking this apart. right? You probably ate some protein today. Maybe you ate beans or chicken or something. You broke apart the proteins inside that chicken or those beans, and then you're going to rearrange those amino acids into something that you need for your body. Okay, what's the secondary structure? Well, due to hydrogen bonding, this is pretty cool, right? Between O's and H's, between different amino and carboxyl groups, I'm using terminology that's too confusing. Between this part and this part in different ones, there's going to be hydrogen bonding. So they're going to give them certain shapes, alpha helices and beta pleated sheets. All right, and so we will model that in class if we haven't already. We'll do that kind of off of a Bozeman science video with protein rods. All right, and so they form these three-dimensional shapes based upon their R groups. So now the R groups start interacting, right? The ones that are hydrophobic or that don't want to be near water will go towards the inside of the protein. The ones that are hydrophilic or that want to be on the outside will go to the outside. Some of them have sulfur, and those are cysteine amino acids, and they like to bond to each other, so they're going to make disulfide bridges, which is pretty cool. And so and you can have acids and bases as well. All right, and then you can have multiple proteins come together like hemoglobin. We watched a little video on it, and so that's 4 different protein chains coming together, and that's called quaternary structure, or the 4th level. So what's important from this? Well, DNA is going to code for your protein, so that's pretty important. A change in the amino acids can change the structure of a protein, which changes its function. If that's a good function, it could be something selected for by evolution. So it could be bad. It could be something like sickle cell, but maybe that could be good in areas with malaria, and we'll get into all that. But basically what I would like for you to get out of it, and sorry, the video's been a little disjointed for this section, is that the different structures or different sequences of amino acids can lead to different protein shapes, right? Which gives them different functions. So they gain their function from the way they fold. They help to act as enzymes. They help to lower activation energy for reactions, etc. Okay, we ran tight on DNA and RNA and ATP and nucleotides, etc. So this is just briefly covered, and I'll tell you exactly in class kind of what you need to know. So these are my kids, right? So how did I pass on my genes to them, right? Via DNA. And so what are the functions of DNA? What is the structure of DNA? And how does the structure lead to its function? We're going to do a whole unit on DNA, and that's the beauty of molecular biology. And we'll really get into it. So this is just a nice little preview. All right, so you know that the functions of DNA are to pass on genetic information. So let's look at the structure of it a little bit. It's made up of nucleotides, and nucleotides are phosphate sugar backbones with a nitrogenous base right here. So this is how we figured out that DNA is different than proteins, because it has phosphorus in it. We did that with some labeling experiments, which is really cool. We can put these together, and once again, it's a dehydration synthesis reaction, and we can link together many of these. Right? So genes can be thousands of these letters long. Now, these aren't actual letters in your body. They stand for molecules, right? So this is what an adenine molecule looks like. Adenine and guanine, the G, are called purines, and T and C are the pyrimidines. And A's and T's always pair together, and G's and C's always pair together. And it's because of the hydrogen bonding that can happen between those base pairs. So this is pretty cool. It shows a way that information can be replicated by copying DNA. We'll get way more into that with Rosalind Franklin and discovery of DNA and copying it. So that'll be cool when it's all done with enzymes. Okay. And you know what's pretty cool is ATP is also a nucleotide. People don't realize that, right? And it just has 3 phosphates there. All right, it's just like salt and caboose highway. Traffic runs in opposite directions, so it gives DNA directionality. It's called anti-parallel, which is interesting, but that's the way that it can fit together so it has the appropriate hydrogen bonding. They're linked together through dehydration synthesis. Don't worry anymore about this. We're going to get into this after the winter holidays, and it'll be a lot of fun, and we'll do a lot of modeling, and we'll do a big storyboard and storytelling with it. All right, students, that was it. I hope this video helped you and that you're getting excited about macromolecules. This is not the end of macromolecules because we'll talk about them as we get into cells and cell transport. Good luck studying, and there's one more video to come with enzymes.